Nikki, you kind of answered your own question. You're correct. It does not take ATP for calcium to bind to troponin. The troponin just has a calcium binding site on it, which means it's like a space on that protein that has a really high attraction or affinity for calcium. So you're right. As soon as the calcium gets released from the reticulum and it's just diffusing through the sarcoplasm, it's going to be attracted to the troponin because of that binding site that has that high affinity. So if it bumps into it, it's going to stick to it, and that's what's then going to initiate that cycle. So yeah, you're absolutely correct. That specific step does not require ATP. Christina, you asked where is skeletal muscle located relative to the skin. This is a really easy one-word answer. Anybody want to throw it in? If we go back to our terminology from chapter 1. Yeah, deep compared to superficial. Exactly. That's all that's asking you to do is use that terminology that we learned back in chapter 1. Yeah. So yeah, you could say it's below the skin, and yeah, it's going to be attached to the bone. What would it be compared to the bone? Yeah. What would it be compared to the bone if you're still using those amputee terms? Yeah, it would be superficial to the bone, deep compared to the skin. So that's just trying to figure out where it is in the context. I'm going to scroll back up and keep answering these questions that were put in first before we get into the more recent ones. Megan, you asked about the refractory period, summation, tetanus treat. We'll get to that in a little bit because that's more of an intensive topic. I'll just do the short ones first. Would actin and myosin be an organic compound of protein? So this is kind of two questions, but yeah, the actin and myosin. That's a good question for you guys. Where are they on the levels of organization on that scale of levels of organization? Are they cells? Are they tissues? Are they chemicals? Any ideas? Yeah, they'd be on the chemical level of organization because they are protein. So yeah, actin and myosin, they are types of protein. So yeah, under that macromolecule of protein, that's a good point because protein, when we study proteins in that macromolecules chapter, we don't really go into huge numbers of examples, but this just goes to show that there's a huge variety of protein shapes that you can have. You know, a typical protein we think of like a little kind of round-ish thing, but no, proteins can be fibrous, like in terms of, you know, they can have like a thread-like appearance. That's like collagen. Collagen is a protein that has a fibrous appearance. They can be globular, meaning like round, like hemoglobin or myoglobin. You can have all kinds of shapes of proteins. But yeah, actin and myosin are proteins. They are, if you zoomed in on them, they would be made up of amino acids linked together that are then folded into a specific shape. So yeah, they are proteins, which means they are organic. Okay. Christina asked, deep fascia is made up of what type of tissue? Anybody remember that from the very beginning of the chapter? It's dense irregular tissue. That is correct. That's the tissue type. Let me ask you guys, what's the other name for the deep fascia? It's the dense irregular tissue that covers the muscle. It's the dense irregular connective tissue that actually covers the muscle. And what did we say another name for that is? Not periosteum, epimysium. There we go. You're on the right track, Tessa. It's a similar concept, but that's for the bones. For the muscle, we call that the epimysium. It's the dense, irregular connective tissue that actually covers the muscle. And you can call it deep fascia, or you can call it epimysium. Same thing. Okay. Okay, why is rigor mortis not permanent condition in cadavers? So let's go back to talking about what rigor mortis is. If we think about the what is it? The cycle of actin and myosin binding and then unbinding during the sliding filament model, a lack of which chemical causes rigor mortis? That's a hard wording. It's hard to put words together, but yeah, thanks, Megan. You picked up what I was putting down. That the ATP is needed to keep that cycle going. If you don't have ATP, then myosin will never unbind from actin, because in order for this full contraction to happen, myosin has to bind to actin, pull it, then let go, and bind to a new spot, right? Like pulling a rope hand over hand. The ATP is required to unbind myosin. So if you don't have ATP, then myosin just sort of freezes, holding on to the actin, and that's why the muscles freeze up, and that's what rigor mortis is. But it doesn't last forever. It only lasts for, like 12 hours, maybe a day or so, because what starts to happen in the body at that point? Yeah, decomposition, decay. The proteins are just going to start to break down, just as a natural part of decay. So the actin and the myosin technically the cross bridge will break apart, but not because ATP is binding to it. It's just because those proteins are actually breaking down. HJ asks, ATP comes from calcium. No, ATP and calcium are two separate things. The ATP is needed in that contraction cycle after the calcium. Sorry, we've got double meetings going on in my house. Let me scroll back up to where we were. So just keeping down the line, Sheen asked, when does the muscle contraction stop? What stops the contraction? So let's go back and think, what caused the contraction in the first place? What initiated the actual contraction, if you go all the way back to the beginning? An action potential. So an action potential was being sent by the neuron. So that's what initiated the contraction. So to stop it, the action potential has to stop. So yeah, as soon as what stops the contraction is the action potential, and the neuron stops firing. And that's going to cause like a whole chain reaction, right? Because if the action potential isn't firing anymore, then you're not going to be sending the neurotransmitter across. That means that you're not going to have the action potential going on in the muscle cell anymore. And it's all just sort of going to work in reverse. Okay. Sure, we can relate it to sodium. Let me just actually pull up the... Yeah, this is a good chance. This is a good time for us to just go through the... Sorry, this is all of our chapter 9 stuff. This is a good time for us to just go through the neuromuscular junction. So, you're correct, Megan. But let's start by kind of talking through what has happened. We sent the action potential down the neuron. We released the acetylcholine. The acetylcholine bound to these chemically gated sodium channels on this side. And if the chemically gated sodium channels opened, what are they allowing into the muscle? Nope, not calcium. They're letting in the sodium, because that's an action potential, right? When you let sodium in, sodium has a positive charge. So if you're letting in huge amounts of sodium into the interior of the cell, you're allowing positive charges in. That's flipping the charge of the membrane, and that's what an action potential is. So that action of letting in the sodium restarts our action potential. It went down our T tubules. That allowed these voltage-gated calcium channels to open. Our calcium went out, and it went over to the troponin, bound to the troponin, and then we started our muscle contraction. I left out a couple of steps, but you get the picture. So in the reverse order, as soon as the action potential stops being sent down that neuron, you're going to start you're going to close off the release of the neurotransmitter. There's already some neurotransmitter here that's bound, so we have to take that off of these channels, right? Because as long as the acetylcholine is bound to these gates, they're going to stay open. They're the key that leaves the gate open. So Megan's absolutely correct. We have to take them off, and that's done by that enzyme. Does anybody remember the name of the enzyme? Cholinesterase. I'm going to type it as ACH, AIDS here. And that's what takes the acetylcholine off of these channels, which allows them to close, so sodium's not coming in anymore, and you're not having an action potential anymore. If you don't have an action potential going down the T tubules, then our calcium channels are also going to close. They're going to close, 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 and that's when these guys, these calcium pumps, work to pull the calcium back into the sarcoplasmic reticulum. Okay? Because the contraction doesn't actually stop until the calcium unbinds from the troponin. As long as calcium's bound to the troponin, myosin is going to be binding to the actin, because the troponin is pulling the tropomyosin off of those binding sites. It's holding them back so that the binding sites are exposed and myosin can grab onto the actin. So until the calcium actually gets removed from the troponin by the action of these pumps in the sarcoplasmic reticulum, the muscle contraction is actually going to continue. So if you didn't have these calcium pumps, or if these calcium pumps weren't working, let's say they've got some kind of mutation in them or some kind of thing that's stopping them from working, what's going to happen to your muscle if these calcium pumps didn't work? Well, if these calcium pumps in the sarcoplasmic reticulum are not working, you're not taking calcium off of troponin. Calcium's going to stay stuck to the troponin. So Nikki's correct. As long as the calcium's stuck to the troponin, it's going to keep contracting. Yes, y'all are correct. If these calcium pumps, because the purpose of these pumps is to push calcium back into the sarcoplasmic reticulum, to take it out of the sarcoplasm, which means it's going to unbind from our little troponin here. Yeah, these pumps, they're pushing the calcium back in. So if they aren't working, calcium stays stuck to the troponin, and the muscle will keep contracting. Same thing if you didn't have this enzyme, this acetylcholinesterase. If that enzyme was broken, then the acetylcholine is going to stay present to these channels, which means the action potential is just going to keep going, which is going to cause these channels to stay open. So yeah, there's a couple of steps that you could interrupt that would result in the same thing. Yes, Megan, you're 100% correct. Since these are pumps, anything that's a pump in your cell is something that requires ATP, because it's pushing the calcium against its gradient. There's already calcium in here, and the pump is forcing more calcium in to the sarcoplasmic reticulum. So since we're going against the concentration gradient, it requires energy. Okay, I think I hit a couple of people's questions. Let me scroll back up. Tessa, way back, you asked if the calcium rushes into the sarcoplasmin that doesn't use ATP. Correct, yeah. So this step where the action potential opens these gates, because you already have a calcium gradient in here because of these pumps. These pumps, again, they're pushing calcium into the sarcoplasmic reticulum, which means you have a super high concentration of calcium inside the SR. So if you open the doors, all that calcium is going to want to rush out. It's going down its concentration gradient. That's diffusion, going from high to low. That does not require energy. The thing that requires energy is these pumps to push the calcium back in, because that's against the gradient from low to high. Okay, let me go back. Okay. So, Shane, you asked about atrophy and hypertrophy in the muscle. They're both at the cellular level. You're correct, because what they are is the cell either growing or shrinking in size. Anybody want to take a guess as to what's actually gonna? Let's start with hypertrophy. What can you do to make a muscle cell bigger? What are you gonna have more of inside of it to cause hypertrophy? I know we kind of changed gears a little bit. Mitochondria is not really going to affect the size of the cell because mitochondria is a relatively small organelle. More calcium will make it bigger, because if you have more calcium, what are you going to have a bigger one of if there's more calcium to store? I'm kind of hinting by circling it on the drop down. Yeah, okay. So if you have more calcium, you're going to have a bigger sarcoplasmic reticulum in the muscle cell, and that is going to make the muscle cell bigger. So that's one way to make the cell bigger. McHenry, you answered the other way to make the cell bigger is to just have more myofibrils. So myofibrils are the protein here, or well, they're the actin and the myosin, the thick and thin filaments that are made of protein. The myofibril. Let me just draw a line around it so we can kind of figure out what it looks like. Remember, the myofibrils are the kind of units inside the muscle cell that are broken up into sarcomeres. So one single muscle cell will probably have, I'm just going to throw a number out, like 20 to 50 myofibrils inside of it. And these are the things that actually get shorter during the muscle contraction inside the cell. So if you have more myofibrils with more protein, your cell's going to be bigger. It's also going to be stronger, because the more myofibrils you have, the more space you have basically for contraction to happen, true contraction, and that's what makes your muscle stronger, and that's why you need a lot of protein to make your muscle stronger. Yeah, more fibers is one way to think of it, but yeah, it's really like in a single myofibril, you're going to have a set number of filaments. So what you're really going to do is add more myofibrils. Remember, this whole thing is a myofibril. These individual things are the filaments. Okay. So yeah, that's two ways to do hypertrophy. Bigger sarcoplasmic reticulum, because that's a really big thing that takes up a lot of space in the cell. So if you make it bigger, the whole cell will get bigger. Or more myofibrils. All right. A couple of you guys mentioned back the diffusion does not require ATP. That's correct. Diffusion, any kind of diffusion, passive, like simple diffusion, facilitated diffusion, osmosis, those are all forms of passive transport. They do not require ATP. Did I miss any questions? I think we're caught up now. Any other questions about this topic before we jump to another muscle physiology topic? You guys are good? Cool. Well, let's change gears a little bit. That's just a question. Yeah, go ahead, Tessa. I'm sorry.
Okay, my question is with the voltage gated versus just the chemically gated channels. Can you go over that kind of?
Yeah.
Because I know one of them you can't have I think it's voltage gated that starts it, and then from thereafter they're chemically gated. Did I have that in the right order?
Well, it depends. The voltage gated and chemically gated, they're just two different kinds of channel that you can have. So the way those channels are named is for how they're opened and then what they let in. Okay, so for example, these are voltage gated calcium channels. So the thing that opens them is an action potential, because that's our voltage change.
Right.
And the thing they let in is calcium. These guys are chemically gated sodium channels. So they are opened by a chemical, which is our acetylcholine, and the thing they let in is sodium.
Okay.
Those are the only two we're dealing with here. When we move on to chapter 12, you're going to see that we also have voltage gated sodium channels. So those would be ones that are also opened by an action potential, but they let in sodium. There's voltage gated potassium channels. So it's not necessarily about an order that they're occurring in. It's just about what they are opened by and then what they let in.
Okay.
Does that help?
Yes. That section still kind of confuses me, as how to tell the difference. If they ask the sodium gated on the motor end plate, is that voltage gated or chemically?
Well, it's not. These are not sodium gated. These are chemical gated sodium channels.
Okay, so that's why I was that's where my confusion lies, I guess. I don't have to.
Yeah. For that it's not really a matter of being able to tell. It's more, that's just something that you have to remember, that's where it's located. Because I'm about to make things more confusing for you. Once we learn about what actually causes an action potential, you're going to learn that right here at the motor end plate, you have these chemically gated sodium channels, but then right here for the rest of the sarcolemma, you have voltage gated sodium channels and voltage gated potassium channels, because those are what are necessary to actually cause an action potential to happen. So you've got all kinds of channels present in the membrane. We're just focusing on these right now because these are what are relevant to this physiology process.
Oh, okay. All right. Thank you.
Mm-hmm. Yeah. And it kind of makes sense that, and this is just for everybody, it kind of makes sense that you would need chemically gated ones right here, because the action potential can't jump across this space. We have this synaptic cleft here. So it wouldn't make sense to have voltage gated channels here because there's no way for the voltage to reach them. So you have to have chemically gated ones on the other end of the neuron, because the only way to kind of keep the signal traveling is to release this chemical here. Because again, the action potential can't just jump across free space. That's a good question, though. So, Megan, you asked, is the calcium leaving the sarcoplasmic reticulum a voltage-gated channel? Yes, it is. These are more voltage-gated calcium channels. So the voltage, they're able to be triggered by the action potential going down the T tubule just by virtue of the fact that they're right next to each other. This picture doesn't show it too well because we leave space to be able to draw things in, but in an actual cell, the T-tubule is like basically touching the sarcoplasm, the sarcoplasmic reticulum. So the action potential traveling down it triggers these to open. Okay. Okay, if he asks, can we talk about the positive and negative charges at the axon terminal? Yeah, the main thing to remember, this kind of goes back to chapter 3 when we learned about the voltage at just the basic state of any cell in the body. Any cell in the body that you look at is going to have a slight negative charge right inside the membrane and a slight positive charge on the right outside of the membrane. And that's just because of the difference between the extracellular and the intracellular fluid. Remember, we had those sodium-potassium pumps that are constantly pushing sodium out at a faster rate than they're pulling potassium in. So that means that you've got more positive charges outside than you have inside. So that's all these little charges mean, is it's more positive outside of the cell than inside. And that's just normal for the cell. That's any cell that you look at the body at any given time. What that means for doing an action potential is that when we want to do an action potential, we want to flip that charge temporarily, right? We want to make it slightly positive inside for a second and slightly negative outside. And the thing that accomplishes that is the sodium, which has a positive charge, entering the cell. Because if you open these channels and allow a bunch of sodium to rush across the membrane, you're making it a little bit positive right here. And that's what the action potential is where you flipped. Where it used to be positive outside, negative inside, now suddenly it's positive inside, negative outside. And that's the signal that then travels down the membrane. Does that answer the question?
Yeah, the answer is indeed, but the synaptic bulb, is there any positive and negative charge on that bulb, at the beginning, at the entrance of the bulb?
Not necessarily. It's the reason the bulb does, the reason why exocytosis happens,
not the exercise, not the ACL place, at the beginning, at the entrance. Yes. At that entrance.
Thank you. This is also a cell. It's a weird shape, but this axon is a neuron, which is it's a type of cell. So this exact same differential is in this cell too. We've got a slight negative charge inside and a slight positive charge outside. So I know it looks strange, but it is a cell too. And so that's why we're able to have this action potential traveling down this membrane as well. Okay.
Thank you.
HJ asks, does the acetylcholine help calcium contract? The acetylcholine is the neurotransmitter in here, right? That's what's inside the bubble, with the little synaptic vesicle. So the acetylcholine is what triggers the action potential in the muscle cell, which eventually is going to lead to the calcium being released. But it's not like it's not directly tied together. No. The acetylcholine causes an action potential in the muscle cell, which causes calcium to be released, which is the thing that actually causes the contraction, because the calcium has to bind to the troponin, and that's what causes the actual contraction to start. Okay. Other questions about this while we're here? Calcium is also positive. Yeah, you're right. Calcium, but the calcium entering here it's separate from the action potential. Yeah, it does get kind of confusing. I wouldn't think too much about it. to keep your brain from kind of exploding. But the thing that you kind of need to just remember is that an action potential is synonymous. It's the same thing as saying sodium's entering the cell. So when sodium enters the cell, an action potential is associated with sodium specifically. I'll just leave it there for now. Sodium specifically entering the cell is what causes an action potential. Okay. Good. These are good questions. Okay. Any other questions about? It doesn't have to be about the neuromuscular junction. It can be about other muscle concepts. So a sodium channel is made of protein. So when we say protein channel, all we mean by that is that it, like, if you actually looked at what this is made of, it would be made of protein. But we still call it a sodium channel because it's for sodium. It allows the sodium in. Oh yeah, okay, we can do that. Thanks for reminding me, Tessa. The treat versus summation, all that stuff. Let me share the whiteboard for a second. So the whole summation, treat, tetanus, it's you can only really see them where we're kind of shifting how we're looking at what's happening in the muscle right now. This chart that I'm going to draw is showing, okay, there's an action potential, and then on top, it's going to show what happens in the muscle. Okay, so if I have that. The way these two things are related is that an action potential happens, and then this tension increase in the muscle happens, or a contraction happens. And She, you're correct. To go back to that, it travels by the sarcolemma, and the T-tubule is part of the sarcolemma. The T-tubule is just a specific part of the cell membrane where it goes into the cell. But yeah, it travels that way. So good. So the thing with like summation and TREAP is that, oh, I'm sorry, let me go back to explaining what this, if you just have one action potential going down one neuron, the resulting contraction of that motor unit is called a twitch. All right, it gets named that because that's pretty much what happens if you have like a twitch in your facial muscles, just like a little bitty little blip, and you're like, above your eyebrow or under your eye. That's caused by just like an errant action potential being sent down that motor unit. So just a little response in the muscle. What happens is when you start sending action potentials, different color. When you start sending the action potentials closer together, you're not giving the muscle cell time to fully relax. So let's remember what's happening here. When you send the action potential, the muscle cell has to receive the action potential. It's got to have the acetylcholine bind to it. It's got to open up its own action potential, open its sarcoplasmic reticulum, allow the calcium out, and then once the calcium starts binding, that's when you actually start to see the contraction happening. This is when the calcium's binding to troponin. And then what causes the relaxation? Calcium unbinding, right? During this whole stage, calcium is unbinding and it's going back into the sarcoplasmic you get back to the muscle being totally at rest, it means all the calcium's back where it belongs. But if you send another action potential before that can happen, then you're opening the doors to the sarcoplasmic reticulum before all of the calcium's been reabsorbed. So you're basically letting more calcium out. So what you would see is here's our initial contraction. And then if you do it immediately again, you might have just a little bit of a bigger contraction. It's just a little bit stronger because there's a little bit more calcium present in the sarcoplasm now. And this, technically, what I've just drawn is TREAP. When you send an action potential immediately after, the summation is if you start sending the action potentials even closer together. Let's do this, two like that. That means that before the relaxation is even done, you send another one, and that's going to be a little bit bigger. There's not a huge difference between TREAP and summation. Really, the only difference is that this is if you re-stimulate it in the middle of it relaxing. No, Jean, there would not be a picture like this where it asks you. All they would ask you is kind of to say what causes it. All right, so why is the contraction a little bit stronger when you send the action potentials closer together? Yeah, TREAP technically has a complete relaxation, and Megan's right though that there's more. The reason why it's a little bit stronger is that there's more calcium left in the sarcoplasm. So that's what you need to know about these things. It's not necessarily a bad thing. It's just that it's like that's one way that you can make a stronger contraction is because when you start sending the action potentials so close together and you don't stop, let's do this, a bunch of action potentials one after the other, that's when you have the muscle just kind of fuse into a tetanus. And I'll do it like this because you still are going to have muscle fatigue. But this is like maximum contraction for the muscle. The action potentials keep coming. So the sarcoplasmic reticulum basically doesn't close. The calcium just keeps leaving. Like as much calcium that can leave, does leave. So every myosin head is engaged with the actin, and you're having a maximum contraction in the muscle. Right, so the reason why we definitely don't want this to happen in the heart, because if your heart is just kind of seized up, it's definitely not going to be pumping oxygen efficiently. So what happens in the heart that prevents you from being able to do this tetanus or TREAP or summation for that matter? Anybody remember the word for it? Not necessarily intercalated disc. It's something that happens during this cycle of the muscle twitch. We had contraction and relaxation, but there's another, the refractory period. That's the word I was looking for. Thanks. So the refractory period is something that happens in the heart after action potential occurs. So I just sort of drawn it in purple here. In the heart, we have a really long refractory period, which means that you can't do like this. You can't have two action potentials sent back to back that would cause a tetanus. Instead, you've got this long relaxation or refractory period where another action potential can't happen. And that means that it's spaced out enough that the next action potential will cause, an efficient beat of the heart. So this refractory period happens so that another action potential can't. So that they're properly spaced out to allow for full relaxation. Does that make sense? Okay. In skeletal muscle, you don't really have a refractory period, and that's why you can have a continuous contraction at the muscle cell. The reason why it has a longer refractory period is not important for y'all to know. So the point right now that you need to know is just that there is a long refractory period where you can't have another action potential. It's not quite ANP. Well, yeah, that is. The actual heart cycle is ANP2. The reason why there's a refractory period is because of something called hyperpolarization that happens in the cell. Okay, let me scroll back up. If he asks, is that an example of treat? No, treat happens when you are stimulating them closer together. So it's you don't really have a refractory period in between, and that's why you have the second stronger contraction. So that would not allow for a refractory period. These are great questions. Anything else about these? Twitch, treat, summation, tetanus. The main thing to remember is just what's calcium doing during them. Cool. All right. Other concepts that you guys want to run through before we?
Yes, can I ask a question, please?
Yeah.
So during the summation and what's the calcium doing when there's a partial relaxation?
Okay, so yeah, the calcium is what's causing the contraction and then the relaxation. So during this half of the kind of twitch cycle, the calcium is being released from the sarcoplasmic reticulum, which means that it's binding to the troponin, which is causing the actual contraction in the muscle. And then as the calcium gets taken away from the troponin and reabsorbed into the sarcoplasmic reticulum, the muscle starts to relax. It's kind of a slow process, relatively slow, that the calcium kind of gets pulled off a little at a time. And that's why you see this kind of gradual decrease. So what happens in these two is that you re-stimulate them, which means that you're reopening the sarcoplasmic reticulum before all of the calcium could get reabsorbed. And that means that the next time you open the doors and allow more calcium out, you have an overall higher concentration of calcium present in the muscle, which means that more of a contraction is able to happen in these two. Because you reopened the doors to the sarcoplasmic reticulum before you put all the calcium back inside of it. Okay. Great. Anything else? I have 5 review questions for you guys from this unit that we can do if there's no other questions. Okay. So on this one, you guys can actually draw on the board a circle which ones apply. There's multiple correct answers here. So which sections of the sarcomere get smaller during contraction? Actually circle the answers. Feeling pretty confident about A and C, that the I band and the H zone are the ones that get smaller, and you guys are correct. The inline is not it's not a zone that can shrink. It's a line right down the middle, just like the Z line is the lines at the edge of the sarcomere. So while the Z line moves, it doesn't actually physically get smaller. Same with the N line. The N line is just the middle point of the sarcomere. But the ones that actually change size are the I band and the H zone. They get narrower as the contraction happens. Make sense? Okay. The movement of what ion into the cell is associated with an action potential? This is just a short answer. You can write it on the board or you can type it in the chat. Yeah. Sodium, exactly. When sodium enters the cell, an action potential begins. That applies specifically in the muscle cell during this chapter. We open the sodium channels in the motor end plate, and that starts the action potential in the muscle. Great. This is another one where there's multiple correct answers. You're right, Aisha, that potassium is going to leave during an action potential, but that's something that we're going to look at in chapter 12 a little bit more. But yeah, you're on the right track. Okay, this one has two answers. Which two actions are calcium ions responsible for? I'll say directly responsible for, to clear up any confusion. Because I see that lots of people circling A and C. A couple people did D. Correct answers are A and C. So calcium binding to troponin is what initiates that cross-bridge cycle. That's one of the most important things. The other thing that calcium does during this physiology process is that when it enters the axon terminal, that's what causes the acetylcholine to be released. So remember, that's why we have those voltage-gated calcium channels in the axon terminal. So the action potential hits the end of the axon, opens those voltage-gated calcium channels, calcium goes into the axon, and that's what causes the acetylcholine to then be released. Calcium goes in, acetylcholine goes out. Okay, so that's one role of calcium that we tend to kind of forget because we're so focused on it, on this job that it does. But there's two things. Okay? And it's not directly responsible for causing an action potential, because what it does is it allows the acetylcholine to be released, and then the acetylcholine drifting across, binding to those voltage-gated sodium channels. That's what starts the action potential in the muscle. The cross-bridge cycle is what happens between actin and myosin. That's just a different name for it. It's where the myosin binds to actin. That's what a cross-bridge is the myosin grabbing onto the actin. And then it pulls it, and then it lets it go, and then it rebinds to it in a new place and pulls it and lets it go. That's what it's called. Because the cross bridge is the word for the myosin binding to actin. Okay. Another multiple answer one. When is ATP needed during the contraction process? There are three answers here. Okay. I see lots of people circling B. It's not B. That is not something that actually requires ATP. That's why I put this one in here. Okay, let's talk about ATP. A lot of people put A. Yeah, that's correct. It's responsible for releasing acetylcholine because that's just exocytosis. And then a lot of people put E, which is also correct, that it's needed to run those ion pumps to put everything back to normal after the action potential and contraction have happened. But let's talk about B and C. And to think about this, you have to change how you think about myosin. Myosin is not necessarily, you can't really think about it as like a little arm that grabs onto actin and then pulls it. You have to think about it more as like a catapult or like a slingshot, okay? Because all of the energy into setting up that myosin goes into this step. Okay, ATP binding to myosin is what causes it to let go of actin. And then ATP getting hydrolyzed, which is where you're actually transferring the energy to myosin, that's what requires energy, okay? Resetting the myosin head. I don't have a diagram here, but if you imagine like a little catapult that's supposed to swing, that's sort of what myosin, sorry, get it better. There's our actin, here's our thick filament. So if you're imagining the myosin as being like a catapult, all of the energy goes into setting a catapult, right? It goes into when you pull it back and you like spring load it and push it down into its kind of like locked and ready position. That's where it actually requires you to put force into it. So this step is what requires ATP to be hydrolyzed. Once myosin binds to actin, pushing forward the rest of the way, that power stroke, doesn't require energy because that's like releasing the catapult or letting go of the slingshot. It just kind of shoots forward at that point because it's spring loaded. Okay? Does that, thinking about it that way, help a little bit better? This actual power stroke does not require ATP to be hydrolyzed. Okay. That changed my whole perspective of this chapter when I started thinking about it as a slingshot rather than as like a protein that pulls. Let's see, what is this? So if you just kind of repeat it, if you're thinking about this myosin, here's our little golf club of myosin. As a, if you know what like catapult is, or like a I don't even know another word for it. The things that like throw rocks, trebuchet, I don't know. It's like a spring-loaded thing that you push down against a spring, and then when you let go of it, it springs forward and it pulls it forward. So when you think about myosin that way, all of the energy has to go into loading it. Yeah, catapult is a good word for it. And yeah, the ATP is used to pull it down and load it and reset it into that ready position. So that's when we use the ATP. Once it binds to the actin, that's the trigger that releases it. So then it can swing forward. It can take all that energy that was stored and push it forward. Okay. Yeah, it's kind of a two-step process, Megan. You release it, you unbind it, and then you push it down. So the ATP binding is what causes it to let go of the actin, and then the ATP getting hydrolyzed is what pushes it, pulling it down, yeah, like reloading it. Yeah, pivot, detach, reload is a good three-step thing. I don't have the lecture notes in front of me, but I would assume that the wording is something along the lines of like resetting the myosin head or recocking the myosin head. Detaching the myosin does require ATP, and the ATP just has to bind to it, okay? So something about proteins is that when things bind to them, they kind of change shape a little bit. So when myosin is bound to the actin, it has a particular shape. It's like it's grabbing onto it. When ATP comes along and also binds to the myosin, it causes it to change shape. in such a way that it lets go of the actin. So the ATP doesn't get used up during that step. Just the fact that it bound to the myosin causes it to let go of the actin. You can kind of think of it as like the myosin can only be bound to one thing at a time. So if it's binding to ATP, it has to let go of actin. And then when I'm talking about the ATP being used, I'm talking about like the phosphate getting broken off and the energy being released. Yeah. So that hydrolyzing, breaking the phosphate off and using the energy is what does this actual step where it loads back to get ready for the next cycle. So it's you're correct, Iffy, that binding doesn't require ATP because the myosin has an affinity for the actin, but unbinding does require the ATP. Because otherwise it's just going to stay stuck to the actin. Does that make a little bit more sense to you guys? Okay. Good. I'm glad we did this question. Yeah, Sheen.
So I'm kind of confused because when I read the lecture note, it's that the ATP needs in muscle contraction. One of them is still power stroke, and it's different. The wording is different what it has on the lecture note. So I'm still confused. So like, ATP is not needed on power stroke at all, or does it still need ATP in power stroke?
What's the exact wording in the lecture notes? Do you have them in front of you?
Yes, I do. So it's the B. It was point B, power stroke. It said the hydrolysis of ATP and then the binding of actin caused the release of P and ADP and the swiveling of the myosin head.
Okay, so what happens there? That's let me pull up my other diagram to explain better. What happens during the power stroke is that the ADP gets kind of kicked off of the myosin head during the power stroke. So let me pull up a picture of it so we can talk about it. Picture, but you guys see on the right hand side of the screen the cross-bridge cycle. So what happens is if we start over here, we see the ATP binding and letting go of the actin, and then we move over to the right and we see that the ATP got hydrolyzed, which means now it's an ADP and a phosphate. Because we moved the energy into the myosin head, and that's what caused it to reload. But it's still stuck to the myosin head. It's still bound to it. So what happens during the power stroke is the myosin binds to the actin while it still has the ATP and phosphate stuck to its head. And then during the power stroke, the ADP unbinds from it. It lets go. Because as the protein moves, again, it's going to change shape and it's going to kick the ADP off. So people think about it in different ways. You can think about it as when the power stroke happens, the ADP gets kicked off of the myosin, or you can think about it as the ADP unbinding causes the power stroke. It doesn't really matter either way. The effect is the same, that during the power stroke, the adenosine diphosphate gets released from the myosin head. So it's not the same as required ATP, because the ADP, that's what's left over from the ADP. The ADP is what's left over from the ATP. All right, like that is the ATP is just adenosine triphosphate, and that's what we had stuck to the head originally. And then it gets, it's kind of a matter of semantics. I have to look at the lecture notes, but you don't hydrolyze. The main point is that you don't hydrolyze ATP during the power stroke, meaning you are not transferring the energy from ATP to the myosin during the power stroke. Okay. ADP, the adenosine diphosphate, is kind of along for the ride during that process. But you are not hydrolyzing ATP during the power stroke. Does that help? Okay. Yeah, I wouldn't. Let me see if I've got the lecture notes. Okay. So I see what you're referring to. So it says the hydrolysis of the power stroke is just the title of that section where it says B power stroke. That's just meaning that's the title of that section. When then what it's saying under that is explaining the hydrolysis of ATP and the binding of actin. Yeah, it is worded confusingly. I see what you mean. I'm going to have to take that up with the next version of this lecture notes. But no, the point is that hydrolysis happens before the power stroke. We'll just end full stop right there. Hydrolysis of ATP happens before the power stroke. That's all. That's all we can say about it for now. Okay. Sorry that, I didn't realize it was worded so confusingly in the lecture notes. Okay, let's go back to where we were. All right, I think I've got one more question for you guys. One other thing to think about during muscle physiology. Layla, you're correct. The power stroke does not. So, okay, never mind. Let's focus on this question that's right here. A muscle that mainly does aerobic respiration to produce ATP is going to have a high amount of what inside of it? All right, you guys all got that. It's D, myoglobin. All right, myoglobin is important because it carries what? So it's holding on to oxygen inside the muscle cell. Hemoglobin is the one that carries oxygen to the muscle in the blood. It's in the bloodstream, carries it to the muscle, and then once that blood vessel kind of passes through the muscle, the myoglobin that's inside the muscle is going to take the oxygen and hold on to it until it